All right, thank you so much, guys, for coming. So today we're doing lecture 2. We're going to talk about basic chemistry. We're going to talk about the compounds in the body and how they form the organic molecules that are responsible for all the metabolic reactions in the body. The carbohydrates, the lipids, the proteins, and the nucleic acid, which are responsible for the genetic material. Okay, and then we'll talk about the structures of those compounds today. The first part of it is just going to be basic chemistry that I left out in terms of matter, compounds, elements, and you're all familiar with that, right? Atomic weight and so forth, and know the difference between mass and weight, right? Okay, what's the difference between mass and weight?
Weight is the gravitational pull.
Yes. And the mass, what is mass? Yeah, it's just anything that takes up space, is mass. All right, anything that takes up space is mass, and the amount of material in it, in that substance, is matter. And then the gravitational pull, of course, is there. And a molecule, 2 atoms together, right? And a compound, you know the difference between a compound and a molecule, right? A compound would have 2 different elements in it, right? Yeah, okay, good. And ionic compounds and covalent compounds, we'll cover a little bit of that. But most of that I'm assuming from Chemistry 101 or something like that, okay? So we can move on. And then I'll kind of try and touch over some of the things that you kind of had challenges with, especially when we get to the organic compounds and how you rank them in terms of the primary structure, and moving on to the quaternary, which is the ultimate tertiary structure there is. Okay, and I'll kind of go a little bit more into detail in that. And if we have some time, we might go through those probes on the LS modules. So, properties of elements, right? We got the physical properties. These are what you can detect with your senses, right? So when we first started the class, we talked about anatomy and physiology, and they fit into one of these 2 things. So the first one, where you talk about physical characteristics, is that referring to the physiology or the anatomy? Yeah, the anatomy. This is anatomy, okay? And it infers about the characteristics, the physical attributes. And then the chemical properties pertain to the way these substances react, atoms react with one another, and to form molecules and bigger compounds and so forth. So this would be the physiology part of things. We're kind of sort of building up all the way from chapter 1 going up to... all right, so very good. So the body is made up of most of the elements that make up the body are made up of the 4 compounds, the 4 organic compounds we talked about, which would be the nitrogen, the oxygen, the hydrogen, and the carbon. All right, so 96% of the body is all those elements. Okay, and this make up the organic compounds. All right, so we got more phosphorus. You will notice as we talk about the organic compounds, we will be starting off with CHO, and then we'll be building up and putting some of the other lesser elements that form about 4% of your body, like phosphorus is found in which of these elements? Phosphorus. Which of these organic compounds do we find phosphorus? Nucleic acids, or carbohydrates, or proteins? Where do we find phosphorus? In which of those 4 organic compounds do we find phosphorus? Yes, nucleic acids. Remember, with the phosphate, the sugar, and the base? All right, where do we find sulfur? In which of those 4 organic compounds do we find sulfur? Sulfur. Sulfur. So we said nucleic acids. What's the other one? What's the matter that, or the compound, the organic compound that makes muscle? Proteins. Proteins, yeah, we find sulfur in proteins. Okay? And then, of course, sodium and potassium are responsible for the action potentials, which we'll be covering shortly. They're responsible for membrane potentials. Iodine, it's also found in some compounds. The thyroid gland makes those, and they're based on tyrosine, which is one of the amino acids, all right? And they regulate metabolism, all right? And so forth. Magnesium, some of these ions are associated with enzymes, all right? So that's about 4%. And then there are other elements, such as zinc and them, that are trace elements, found, or selenium, things like that, found in very trace amounts, and usually they are parts of enzymes that, increase the activity, or, regulate the activity of enzymes, all right? So selenium, cobalt, copper, those are some of the trace elements, all right? Manganese, all right? They are found in less than 0.01% in the body, and they're mostly attached to the enzymes, all right? They're found as part of enzymes required in very minute quantities, all right? So I want you to appreciate these kind of elements that make up the compounds, because they influence the way those compounds work, all right? Okay, so here we go. So the types of chemical bonds, I think we had a few problems with ionic bonds in your LS module. So ionic bonds is when the atoms either lose or gain an electron, all right? So they are positively charged or negatively charged, right? Okay, and then when they've lost or gained an electron, then they attract each other. So the ones that lose an electron are called what? Ones that lose an electron. If you lose an electron, what kind of charge do you have? Cation. If you lose an electron because it's negatively charged, then you're going to be positively charged, and it's a cation, right? Can you give me an example of a cation? Yeah, sodium is my favorite cation. There you go. And then, of course, the other one, is an anion. It's a plus electron, and our favorite example is chloride ions. All right. Okay. So, and therefore they all attract each other. Okay. So that's an ionic, and it's arranged in a lattice. It's well arranged. The ionic compounds are. All right. And then the other one is a covalent bond. What's a covalent bond? What happens in a covalent bond? Yep. They do what now? They share the electrons. Okay. How do they share the? Huh? Yeah, non-metals like carbon and hydrogen and oxygen, and they form covalent bonds. Okay. What kind of covalent bond do we find in oxygen or hydrogen? No, I said what kind of covalent bond? Because there are two kinds of covalent bonds. What's that? Well, yeah, that too. But I'm not. I'm talking in terms of how the electrons are shared. I'm thinking of the elections already. Come on, we're 2 years out. In these compounds, they are equally shared. The electrons are equally shared between the two. So it's a non-polar covalent bond. Okay? So equally shared. So they are non-polar covalent bonds. And then the other ones, they are not equally shared between the two. Then you will have a polar compound, because and these polar compounds occur when you have mostly which compounds? Oxygen, when it's bound to hydrogen, or also you can have nitrogen instead of oxygen, because the electronic pool on the oxygen is much greater than on the hydrogen ions. All right? So because this is missing 2 valency electrons on the outer shell, so it really attracts electrons to itself more. So you end up, whenever you have hydrogen, and hydrogen is only a proton and an electron around it. Okay, so when a hydrogen atom loses an electron, we sometimes even call it just a proton, because that's all it is. It's a proton. All right. So the electrons are pulled here. The 1 electron on hydrogen is pulled towards here. So you get what is called a slight negative charge. Okay, and then you get a slight positive charge on that one. So whenever you have that, you have what are called hydrogen bonds. That's why water molecules kind of attract each other, and then that's a polar covalent bond. Polar covalent bond. That's how you form hydrogen bonds. Okay. In the exam, I also want you to be able to explain these covalent bonds and talk about the polar covalent bonds as well, and how they happen. Okay. So that's hydrogen bonds. So I want you to know that. And then, of course, the covalent bonds, like Bernal was saying, I remember your last name, you have double when you have 2 pairs of electrons shared, or a single covalent bond when you have 1 pair of electrons shared, right? So double is usually annotated like that, and then single like that. Then you have to have H on that other one to finish the valency shell of carbon. It needs 4, right? Okay, very good. So, excellent. So, we get 2 kinds of reactions that occur, right? A synthesis reaction is also called a dehydration reaction, all right? Because the compounds that are joining lose water. So in a synthesis reaction, you form 2 smaller substances to form a much more bigger and complex substance. All right? Okay, so you should be able to explain these reactions. All right? So you lose water, so it's a dehydration reaction. All right? So that's basic. And then the other one is a decomposition reaction. It's a hydrolysis reaction. So hydro, water, lysis, breakdown. You use water to break down a more complex substance into simpler forms. Like when you break a disaccharide into the 2 monosaccharides. All right? So you use water, and then you add water to those compounds. So one of the substances accepts the H plus, and the other one accepts the OH. All right? In this case, you have 2 glucose molecules. What disaccharide is that? That is 2 glucose molecules. Because you can't have problems with disaccharides in your LS module. What disaccharide is that? The one that has 2 glucose molecules? Maltose. Maltose is the disaccharide that has 2 glucose molecules. And then fructose and glucose is sucrose, the one you find in the fruits. The sugar in the fruits is sucrose. It's got fructose and glucose together. And then lactose, the one you find in the milk, it's a disaccharide that has galactose and glucose. Okay? All right, very good. So now we're talking about the rate of the reactions and the certain factors and how they affect rates of reactions. All right? We already know that with our homeostasis, we maintain the body temperature at 37 degrees C, because all the reactions in the body, that's the appropriate temperature. All right? So, concentration. The higher the concentration of the reactants, the the faster the reaction occurs. Why is that? Why is it when you have high concentration, you have faster rates of reaction? Yeah, you're on the right track. There's more. What are we doing when we are reacting something? Are we breaking down or putting together? Yeah, so we're putting together when we are building, right? Because we're joining those, we're reacting those two chemicals, whatever they are. So the more there is, the more chance there is for those atoms to bump into each other and react. You see what I'm saying? So you're on the right track, just not the breaking. What's that? Yeah, we're going to talk about that in a little bit. So yeah, that's good. So the more there is, that means, because they have to come together, right? You know, you have to be together to do something together or to react with each other. Okay? So the more there is, more equals a greater chance of colliding. So that's the reason for that. So in the exam, if I ask you not only to tell me what affects rates of reactions in the body, and I ask you why, how they affect the rate, that's what you'd say. Then you get 2 points. So temperature, chemical reactions get speeded up when the temperature increases. Again, it's the same concept. Why does increase in temperature increase the rate of a reaction? They're moving faster. When they're moving faster, the likelihood of you bumping into each other, that's why the speed limit is 70. I like it to be 80 though. Because if you're going fast, you know, the chances of you reacting and in time to stop it, you bump into each other more. That's basically what it is. So when the atoms are going much faster, the chances of them bumping into each other and reacting is much faster, so the reaction goes faster. All right? Okay? Faster speeds equals greater chance of coming together. Because what you want the elements to do, or whatever you're reacting, your reactants, is to bump into each other and make a reaction. All right? And then, of course, catalysts. Which of the 4 compounds make up catalysts? It may be on there. Yeah, enzymes, yeah. Proteins, the compounds. I was talking about the biological compounds, the organic compounds. Yes, there are enzymes, yes. And enzymes increase the rate of reaction, and they themselves are not changed. Okay? They themselves are not changed or depleted. Why is that important? Why is that important? When an enzyme, why is it very important that the enzyme is itself not altered and is itself not depleted? What's doing the catalyzation here? What's speeding up the reaction? The enzyme, okay? So what do you think would happen if you were finishing it up? There will be nothing. Yeah, if you saw that, you would have to make more enzyme. So it's important that a catalyst, when you describe how a catalyst affects the rate of a reaction, you have to mention that it itself is not changed, because if it's changed, then, you know, because it's very specific for one, it will only attach to a certain compound. You know, that enzyme lock and key thing. If you change the structure of that enzyme, which is a protein, then, you know, it's not going to be able to attach to those reactants anymore. Okay? So you need it for it to remain the same, and you need for it not to be used up. Because then, you know, it will just do it for a little bit and then quit. So that's important. Okay? And these are proteins. Oh. You see, we're using up all those organic chemicals, and I'm going to be talking about them in a little bit. All right? So that's how catalysts increase the rate of reaction. Particle size, again, is the same kind of reasoning. The smaller the particle size, the faster the reaction. Again, yeah, this is a bit to do with colliding, of course. But what else does is another reason why smaller particle size equals higher rates of reaction. Yeah, there's more of it, but we're talking in terms of the actual size, the magnitude of those atoms, the magnitude of the reactants. They are much smaller. Why is being small equals higher rates of reaction? Yeah, they can move faster, but there's another reason why. It's got to do with surface area. Okay? It's got to do, if you have a large, like big ice block, let's put it like that, okay, and then I give you a smaller ice block. If I ask you to melt the big ice block, it takes a lot to go to the deeper ice, but if the smaller block, because it's got the surface area in relation to the volume is almost equal, so you get most of the surface area of that ice block, so it will melt faster. Okay, the reason why the bigger ice block melts slower, which is slow rate of reaction, then is because you can't get to the ice deep in the middle. You can only get the bit of ice on the outside because the volume is much greater than the compound, the area on the compound. You see what I'm saying? On the atom. Can you visualize that? So that's another concept about rate of reaction. The more of the atom is exposed, the faster it will react. If some of the molecule is hidden because it's got layers and layers and layers, it's much bigger, then it will go slower. Okay? So I want you to understand all those different concepts, because each one has got a specific reason why it works out. Okay? And understand it that way. If you understand it like that, you won't forget. If I ask you what are the factors that affect rates of reactions, you just go like that, because you can associate it with the reason. Okay? It seems like learning more, but that's how your brain works in terms of retaining this material. All right, so very good. So now we're talking those organic compounds. These are the molecules in the living organisms, and those are the four. We got the carbohydrates, we got the lipids, we got the proteins, we got the nucleic acids, and all those elements we talked about on the first page make up these organic compounds. All right. So we'll cover each one in turn and talk about the characteristics and some of the functions. At the end, I'll have a nice table that summarizes the functions of all those compounds. And I want us to know those functions. All right? Okay. Carbohydrates. These are compounds made of CHO. CHO, which means carbon, hydrogen, and oxygen, in a ratio 1 to 2 to 1. All right? So when we think about the name carbohydrates, what does this stand for? What? Carbon. Yeah. Okay. What about hydrate? What does that stand for? When I say hydration, what am I talking about? Water. Yeah. It's water. Isn't that the ratio of the three atoms? So that's why carbohydrates are called carbohydrates, because they hydrate with the water part. If the ratio is C to H2O, that's literally what it is. You see what I'm saying? If I do that, CH2O, that's why they're called carbohydrates, because they got the formula results in water plus carbon, carbohydrates. So that's where the name comes from. All right? So it's a major source of cellular food. All right, because what forms the carbohydrates? What's the compound? What's the compound? What's the monosaccharide here that we're talking about that makes up carbohydrates? Sugar, yeah, and which is glucose. Yeah, and cells of the brain only take glucose, nothing else. So you need glucose. That's why people who are diabetic, if they don't manage their glucose properly, they can go into a coma, because the brain will only use glucose. It doesn't use anything else. Okay? All right, so that's major source of cellular food because of the glucose. All right, so we got the monosaccharides, which are glucose, fructose, and then the disaccharides, lactose. And I said lactose is made of what and what? It's got a name that sounds just like it. Okay, let's start with sucrose first. What did we say? Sucrose is made up of the two monosaccharides that make up sucrose. In the fruits, we said fruits. Remember fruits? Fruit. What does fruit sound like? Fructose and glucose make up sucrose. Right? Fructose plus glucose, FG. Okay, fructose plus glucose. All right. And then lactose. We said glucose is one. Okay, what's the other one? It sounds like lactose. Galactose. I know it's a kind of an unusual monosaccharide, but it's galactose. G plus G, okay? Galactose. Okay, I'll spell this one. Galactose. Okay, and then the other one I talked about, maltose, which is glucose plus glucose, is maltose. Okay? And then, of course, polysaccharides are starch. Poly means there's a lot of these monosaccharides. Many, all right? And they form glycogen, which is a store of energy in the muscles and the liver. Muscles and liver, you store glycogen. Why would muscles store glycogen in there? Huh? Yeah, for energy, because muscles use glucose, it breaks down glycogen, especially the white fibers. The white fibers tend to have glycogen in them because then it's just broken down into glucose, and then you get 2 ATP from each glucose when you break it down, all right, into lactate, all right, so polysaccharides, and then starch, of course, they all get broken down. So we have glucose here, and what kind of reaction is this? It's a synthesis reaction, and what else do we call it? Okay, when we think of this, dehydration, because we're giving off the water. Synthesis reactions are dehydration reactions, all right, because we remove water and join the compounds. Hydrolysis means you use water to lyse. It's hydro, and then lysis is the breaking down. So hydrolysis, which is a so you use water to break down, all right. And synthesis, you give off water to join. See what I'm saying, right? And there's just and carbohydrates, another function they have is that they parts of the cell membrane. They extend from the cell membrane for cell recognition, so that cells recognize each other because of the kind of carbohydrate chains that are on top of the proteins or attached to the which are called glycoproteins, because they've got the glycogen from the glucose and glucose chains plus the protein, and those are responsible. They're sometimes called cellular adhesion molecules. So those are found on the surface of cells. So that's another function of carbohydrates, apart from giving energy in the cell. Okay. Cellular recognition. They're sometimes called like selectins, integrins, and so forth. That, for instance, white blood cells, when they go to a site of injury, there are certain compounds, these glycoproteins, that will anchor the white blood cell where the injury is, so that it will sit there and do the job. Otherwise, it will be flowing in blood and it will just move. So the carbohydrates also have that cell recognition, cell adhesion function as well. All right. So we move on to the next organic compound, the lipids. All right. And these don't dissolve in polar compounds like water. Remember, water is polar covalent bond, so it's a polar compound because it's got slight positive and slight negative charge. But they will dissolve in non-polar compounds like acetone and all that, alcohol. All right. So these are hydrophobic, right? They don't like water. Again, we've got CH and O, but oxygen to a much less extent than carbohydrates. Okay, there's very little oxygen in the fats. All right, and the fats are made up of long chains of carbon atoms joined together. All right, and a carboxyl group at the end. There's another thing you guys had problems with in the module, carboxyl group. So it will have like a very long chain like that, and then a carboxyl group. That's a carboxyl group. Okay. Oh, I like the way this thing erases. You know, it only does that part. COOH is a carboxyl group. Okay, carboxyl group. So it's got a long chain. A fatty acid is a fatty acid, and the acid part of it comes from here. What's an acid anyway? From the chemistry. It can give off a proton, a hydrogen ion. Okay, so this COOH can be like that with an O here, and then there's a minus here, and then it gives off the H plus. So it's an acid. Okay. So I won't go into that because that's the chemistry part that I'm assuming you guys have done. Okay. Okay. Very good. All right. So. So that's a long chain, and then they will of course differ, the fatty acids, because they have a long chain, okay, or a short chain. And another way they differ is because they have what kind of bonds in it? Fats. When we talk of unsaturated and saturated fats, what is an unsaturated fat? What is an unsaturated fat? Is it got single bonds or double bonds? Unsaturated. Yeah. It has double bonds. Okay. Because you had problems with that in the LS module again. So fats differ because of the chain, the length of the carbon chain, or whether they are saturated or non-saturated. All right. Non-saturated, would be double bonds, and saturated, it's saturated with the H plus. So it will have no reactivity whatsoever. It will just have C with single bonds on the C, and then H's over here. Okay? But if you have a fat like this, some are monosaturated, some are polyunsaturated. I mean, monounsaturated and polyunsaturated, which means they have more double bonds in it. The polyunsaturated fats are very good, things like linoleic acid and so forth. So we got the fats. All right, or they're also called triglycerides. They're made of the fatty acids on a glycerol backbone. Glycerol is a 3-carbon compound like so. All right, and then it will have H pluses, H is over here. So on this C, there's a protein chain here. On that C, there's not a protein chain, a fatty acid. And then on that C, another fatty acid. You see, they are all different lengths because they're the different fatty acids. And then it's a triglyceride. That's a fat. And then phospholipids, we've seen phospholipids in the plasma membrane, right? And they have got phosphorus on them. And so the phosphorus head, and then 2 fatty acids on them. It's kind of like a modified triglyceride. And instead, it has 2 fats on it and a polar head, which makes the structure of the membrane. All right, so that's phospholipids. And then eicosanoids, these are 20 carbon fats, okay, and things like prostaglandins, arachidonic acid, 20 carbon, and they regulate physiological processes, almost like hormones. All right, and then steroids, they also regulate. What are the famous steroids? That we have, women and men. What steroids do men have? Yeah, testosterone is a steroid. So it's a lipid. And then the female's estrogen, progesterone. Okay, so it regulates reproductive function. All right, steroids. And then the steroids are made from a compound called cholesterol. Cholesterol is not all bad. You need it. You just don't need too much of it. That's all. But cholesterol, all these steroids are based on cholesterol. And it's a 4-ring compound, all right, that will, so it will have, okay, sorry. Okay, and then it will have another, something like that. and then this is a 5-carbon ring, and then the different groups that are attached to it will make up the differences in those compounds. And cholesterol is a longer chain, okay? So those are the steroids, and they are very important for regulating cellular functions, especially reproductive functions. All right, so now we're going to flesh it up a little bit. Fats, of course, they're found in subcutaneous tissues, like subcutaneous fat. It's very important. Even thin individuals have a little bit of fat and subcutaneous fat here. What would that fat do? What is that fat for? Insulation, so you don't lose too much heat. Again, it's part of that homeostatic mechanisms of the body so that you maintain your 37 degrees C. And then when you get too hot or cold, then you can do those homeostatic mechanisms like sweating or shivering to keep it at the same. But the fat helps you to maintain some of that heat, to build it up. Okay, so that's insulation, all right? And also protection and cushioning, like there's a lot of fat behind your eyeballs to cushion it from, your skull, from the skull. And then, of course, phospholipid, we said, is the chief component of the plasma membrane, right? At the end, we'll have like a table that gives you a summary of the functions of all these 4 organic compounds. And then steroids, of course, cholesterol is the mother compound of all these. And then everything else, bile salts, that are also important in fat digestion and emulsification of fat, all right? And then, of course, vitamin D, vitamin A, D, E, and K are fat-soluble vitamins. And then sex hormones, of course, testosterone and, estrogen. And then the adrenal cortical hormones are made by the adrenal gland, like cortisol. Cortisol is another one, and it looks the similar structure to cholesterol. And cortisol can be a stress hormone at times also, if it's in large amounts, because it will increase glucose metabolism. That's why if you are stressed, you gain weight sometimes, because of the stress hormone, that you're releasing too much stress hormone. The cortisol will increase your metabolism, and you start to mobilize more glucose and so forth, and you get fatter. I didn't find a nicer word, but you get bigger, okay. Eicosanoids, things like prostaglandins, these are hormone-like substances actually that regulate rates of chemical reactions, leukotrienes and so forth, and thromboxanes. Thromboxanes are actually important in clotting, all right? They are factors that make the platelets sticky so that you can clot. So they're important in different things, reactions, all the reactions in the body, all right? And leukotrienes are important in the immune system. They activate some of these, white blood cells and so forth. All right? Fat-soluble vitamins, A, D, E, and K. ADEK, I always call it, so you can remember that. Also important in reactions and so forth. And then lipoproteins. Lipoproteins is a lipid plus a protein. And these transport fats in the blood. All right? Make sense? What kind of compound is a protein? Is it soluble in water or insoluble in water? It's soluble in water. So go figure, if you're going to transport fats, you're going to need lipoproteins, because the protein part of the lipoprotein will give it that solubility, because blood is an aqueous environment. It's water. Okay? So you need more high-density lipoproteins. High-density lipoproteins. Okay? Because HDLs, for crying out loud. Okay, HDLs, all right, for that, and a little bit of low density lipoproteins, because the more protein there is, the more soluble it is, the more useful it is in terms of, transporting in blood. Otherwise, the other one will clog up your arteries because it's less soluble. It will tend to settle. All right? Okay, good. And this is just showing you a triglyceride with a glycerol backbone in pink, and then, of course, synthesis reactions, dehydration reactions. All right? You lose water to join the compounds, and you gain water to break the compounds. all right, and then you form a triglyceride. So this is the 3 carbon chains there. They're just drawn equally there, but just because. But they could be different lengths and so forth. All right. So composed of 3 fatty acids, triglycerides are. All right, and then that's the concept we're talking about: saturated and unsaturated. If it's saturated, it means it's got single carbon-carbon bonds throughout. So it's full of like palmitic acid, and you find this in beef, pork, olive oil. And they say you should not eat too much beef or something, because then you get all these saturated fats. These tend to cause cardiovascular problems because they're so unreactive, they tend to just settle, and they're very insoluble. They just settle in there and clog up your arteries. All right? But these ones are better. This one is a polyunsaturated linoleic acids, fish oils, and sunflower oils and so forth, polyunsaturated. So these ones are more beneficial to health. All right? And then, of course, other lipids, the phospholipids have a phosphate head, which is hydrophilic, and the 2 fatty acid chains, which are the hydrophilic. So how are they arranged in a membrane? Isn't it you get this? And then what else you get? Yeah. Why is this arrangement beneficial? Why is the phospholipid arrangement beneficial like that? Why aren't the tails facing outside? Yeah. You expose the hydrophilic part because the cells are bathed in an extracellular fluid. It's all aqueous environment. The inside of the cell, the cytosol, is all aqueous environment. Okay? So you hide the hydrophobic part and you expose the hydrophilic heads. Okay? So it's got to do with the structure, and then it will affect its function and how it works in the body. All right? So these are very important. We'll come across them again when we do the plasma membrane, which we'll do in the next lecture. And then those are, you see, there's the 4-ring structure that is the backbone for all these steroids, and it's from cholesterol. See, this is cholesterol. It's got a longer group over there. And then testosterone, you see the minor differences. It's got methyl groups on it. Testosterone, and estrogen has only got 1 methyl group on it. And then it's got a hydroxyl estrogen. There's not many big differences, and then it's just got an oxygen atom over there. See what I'm saying? But the rest of the molecule is pretty much the same. All right? So those minor differences affect the way it will react and how it reacts. All right? So eicosanoids are like 20 carbon, compounds, and then steroids, of course, are based on cholesterol. They got the 4 interlocking rings, all right? And then they just have different groups on them, for the differences. All right? Okay, proteins. Proteins, these are the ones that form the primary structure, the secondary structure, the tertiary structure, and the quaternary structure, which is the ultimate folding and grouping of these proteins. All right? Okay, so these are macromolecules. They are huge molecules made up of amino acids joined together by peptide bonds. So we are kind of increasing our elements as we go along, right? We got CHO, now we got N in the protein, and we got sulfur. Remember at the beginning I said which one is sulfur, which one is phosphorus? All right. CHO, and we include nitrogen, which is really the distinguishing characteristic for these because of the amine group, NH2. All right? All right? You got a question, Benno? No, you get good. You good. All right, good. There's 20 different kinds of amino acids based on the what is called the R group. All right? An amino acid has got the same basic structure. Okay? All right? So it's got the carboxyl end, all right, and then it's got the amino end, or the amine end, amine. Okay? What makes them different, the amino acids, is what's attached here, what is called the R group. The R group can be as simple as hydrogen. Then you have an amino acid called glycine. All right? If this R group is equal to hydrogen, it's glycine. And then you could make a methyl group over here, and it's alanine. All right? So you just differ that, and that's it. All right? So that's what makes up these 20 different amino acids. And again, if we have 2, then it's a thing, dipeptide, 2 di, and then 3 tripeptide, and then poly for many. All the proteins are polypeptides, right? Polypeptides. Many amino acids, hundreds of them, make up a protein. All right. So, so proteins are polypeptides of hundreds. So the sequence of these amino acids will be the primary structure of the protein. Okay? The sequence. Just the way in which the amino acids are arranged. No folding, nothing like that. Just the sequence of them. This is my shortcut for primary. It's just the sequence of amino acids form the primary structure of the protein. All right? So that's the primary structure. All right. So the building blocks of proteins are called amino acids. All right. And there is the amino acids. I said they include the amine group, which is the NH2 group. They include the carboxyl group, which is the COOH group. All right. And then they contain a hydrogen atom. You see it? So they have the carboxyl group. They have the amine group. They have the hydrogen. And that's it. And then what makes them different is this R group. All right? So the simplest amino acid there is glycine. All right? And then, of course, this one is the one that's found in the thyroid, the one that's associated with iodine, tyrosine. So tyrosine has got a benzene group on it. So it's important, and it's linked to iodine, one of those trace elements that we talked about at the beginning. And it affects metabolic reactions, tyrosine. And they get more complex, you know, and most of the proteins have sulfur as well, all right, in their R group, such as cysteine, methionine. They have sulfur. And the R group, what does the R group, what properties does it give to the amino acids, especially when you put the different amino acids together? The R group influences what? What does the R group influence on the protein chain? Yeah, it's a side chain. So what is it? Why is it important that you have all these different R groups? When there are different R groups, that means they have different properties, right? Okay. And when they have different properties, it means they will attract in a different way. So the sequence of the amino acids affects how the protein folds up to form the secondary structure. That's why if the sequence is wrong, just by one amino acid and everything is off skew, the protein is not going to fold properly. And if the structure is wrong, then it's not going to do the function. If it's an enzyme, remember the enzyme is a lock and key thing, most of which the proteins are enzymes, right? Yeah, so it's not going to work. So it's important to have the proper R group there, because the R group will affect how the protein folds up and form the structure, right? So that's why it's important. So then the functions of the proteins will be like being enzymes, regulation of chemical reactions, right? Structural proteins provide framework too, all right? Like collagen. Collagen, you know, it's a protein. It's a fibrous protein from structure, all right? the muscle contraction, you remember I said muscles are protein, they're all protein. All right. So myosin is a protein, and actin is a protein in muscle, and these are proteins. They are sort of long proteins that slide up on each other like that. So muscle contraction is a protein, is another function of proteins. All right. If I ever ask about the functions of proteins, you just think about all these things, like, what are proteins? Okay, they are enzymes, so they do regulation of reactions. Okay, muscles are proteins, so contraction, things like that. And then you could just come up with all these functions of proteins without even thinking too hard. All right? And then, of course, fibrous proteins, like collagen is the most, ubiquitous protein there is. Some of them are elastic proteins, like in the ear, they stretch. All right? So extended and strand-like proteins that are arranged nicely. Nice, collagen. Some of them are arranged haphazardly, but the strength of collagen comes from a certain arrangement, a specific arrangement of the fibers. So it gives more strength. All right? Keratin is also another one found in the nails. It's a hard protein, very hard protein. All right? And in fact, your skin, your epithelial cells, they are the ones on the top, they're full of keratin. That's why they die off, and then you have a new skin in about 30 or so days, 45 days. All right? It's because of this protein that makes them hard and die off. Okay? Keratin, and then collagen, of course, in the joints, tendons, and so forth. And then the contractile fibers as well. All right? Myosin and actin are fibrous proteins, and they slide against each other like that. And then most of the enzymes are globular proteins. Why? Remember those reasons we talked about at the beginning and how they affect rate of reaction? And we were talking about particle size and so forth. So why is it that globular proteins would be most ideal to act as enzymes? It's to do with one of those reasons we talked about at the back when we were talking about rate of reaction. It's like small particle size, right? Yeah. Smaller particle size, it can mingle in and mix properly. So they tend to be, they're not bulky proteins. Bulky proteins are usually for structure, right? So the globular ones, that's why even immunoglobulins that are the antibodies, they're globulins. That's why they're immunoglobulins. They kind of have a specific shape to them, but they're basically globular. And then they act in immunity and so forth. So antibodies, which are the immunoglobulins I was talking about, hormones, all those enzymes, they tend to be globular because it makes more sense in terms of affecting rates of reactions. All right? Again, like we always said at the beginning, the structure is always related to the function. It's not ever by accident. Whenever something has a certain structure, you better be thinking about some of the functions for that structure because it's going to be related. It's nothing ever is just because, So what is one example of denaturation that we do every single day? Or not every day sometimes. But what is denaturation? What's a prime example? Like we make it in the morning. What's that? Here?
Use the restroom?
No. Well, I want an example of denaturation, protein denaturation. Like when you fry an egg, right? You are denaturing the protein. And you're changing the structure of the protein. And it becomes non-functional after that. Remember, when you crack an egg, it's kind of sort of colorless fluid. When you fry the egg white, which is where most of the protein is, the yellow is most of the fat. All right, there's some protein in there, but the white, crap, the egg becomes white after it's been denatured. Like jelly as well, it's the same thing. When you make jello, it gets denatured and then it sets. So that's what happens. So disruption of the hydrogen bonds, and then the protein is no longer folded the way it's supposed to. And once it's not folded the way it's supposed to, then the structure is not going to work in terms of what that protein does. So it makes it non-functional. Because again, the protein has to be folded a certain way, exact certain way. If it's just wrong, or if one amino acid is wrong, then it will fold incorrectly, and then it will be.
Does that mean that when you cook your egg, obviously you don't make it wrong?
Yeah.
Does that mean it lessens the amount of protein that's in it because it's been
No. The protein is in there. You cannot destroy matter. Remember, you can't create or destroy matter. You can only change it. In this case, you just denature it. But all the amino acids will be there, and you eat it, and it's good. Actually, I eat a bunch of eggs, actually. So you won't destroy the matter. You won't reduce the protein. You just change the structure of it. That's all. And then your body, when you get into the body, what are you going to do to that? It doesn't matter if it's denatured. You're going to break it down into the single amino acids, which is the ultimate denaturation there is, right? You see? Meat, same thing. When you cook meat, you're denaturing the protein in there, okay? One, so that it will be easier to digest, you know. Eating raw meat, like the carnivores, like lions and so forth, they've got a different digestive system that's made up for that. We couldn't eat raw meat because we could, I mean, you know, like sushi and stuff. But beef is even tougher. But the Japanese have this smooth beef that they make, that they also make into, it's called something else, I forget the name of it. But they, you know, most of the time we have to cook it because of that. Because denaturation helps us to digest it properly. All right? So denaturation, that's what it is. It changes the structure, and then it renders the protein in non-functional. All right, so characteristics of enzymes, you know, we kind of sort of covered most of these. You know, they speed up, you know, chemical reactions. And because they lower the activation energy, the energy you need to start the reaction off, which is activation energy. And we talked about how they are globular. And we said this has got to do with small particle size and also ability of movement. You know, the bigger you are, have you seen the athletes who are very fast? They're kind of lean, right? If you're big, you could move fast, but, you know, the smaller you are, the faster you are. So the smaller the protein is, the faster it can move around for one, and, you know, the particle size is small. So it's got all those elements we talked about at the beginning about rates of chemical reactions all rolled into the globular proteins, which makes them effective, right? Are chemically specific. Again, that implies the folding up of the protein. If it's if it's folded wrongly by a little bit, then that lock and key concept, it will not work, because then it won't fit anymore. So it's got to be specific. It's got to be specific. So these characteristics of the enzymes are based on all those functions. These are kind of functional characteristics. Do you see what I'm saying? They are functional characteristics because they affect the function of the compound or of the substance. And this one is just, they're named after usually the substrate. And then ASC at the end. Like lactase will break down lactose into what and what? Yeah, G and G. You got that right. Which one is I'll give you the first G, which is glucose. What's the other G? Galactose. So that's why I brought it up, because, you know, the adaptive learning software, what does it do? If you get something wrong, it will bring it up again in another way until you get it right. So some of you, I'm happy that you stuck with it and you get 5 points. Because if you don't stick with it, you get like 4-point-something points or 3 points. Because there's some guys who get 3 points out of it. Because I make it 3 points or 5 points. I mean, I make it 5 points. So if you don't stick with it, and because the going back to it, that's how you learn it. That's the only way to learn it. So you're actually doing yourself a favor. You're learning it at the same time. When the exam comes, you're done. And then the ASC just shows it's an enzyme. All right. So we will affect these the rates of reactions by controlling 2 characteristics of enzymes, right? The quantity of the enzymes. All right. So concentration. All right. And then the activity of the enzyme. We can slow it down or speed it up. Okay. So that's how you affect the rates of reactions, by altering concentration or the rate of activity of the enzyme. So now, nucleic acids. All right. So you see what we're doing? CHONP. This is more complex, and it's you know, what is actually used for genetic material, the DNA. So the basic is a nucleic acid, and it's made up of nucleotides. It's like a house, but I don't intend it to be. And a base, okay, and a phosphate group. So that's a nucleotide. And the sugar here, the monosaccharide is, it can be ribose, like in RNA, or deoxyribose in DNA. Okay? So the organic bases are things like adenine, thymine, guanine, and cytosine. Okay? Which are the bases in DNA actually. Okay? You can't have problems with that in the LS module. All right? And then uracil, it replaces cytosine in RNA. Okay? Yeah. And we'll go back to it again in the next. So these are them, all right? So these will be DNA, and these actually uracil replaces thymine in RNA. So these are RNA. Okay? And adenine is one carbon, one ring base. Guanine is a two-ring base. That means it's like this. Something like that. Okay, so there's two rings together. And then the adenine and cytosine are just one ring. Okay? So that means the one ring one will pair with the two ring one in the DNA, in the double-stranded DNA. Otherwise, if one ring and one ring, they won't fit because they want to touch. If you have two ring and two ring, they'll be too long. Okay? All right, so 5 nitrogenous bases contribute to the nucleotide structure, but each one has 4. The only difference is that uracil, instead of thymine, is in RNA. Okay? That's that. And then, of course, you have DNA and RNA. Those are the major nucleotides. And DNA is the genetic material that codes it. And then RNA, what does it really do most of the time? What's really the function of RNA? Yeah, to transfer the message and to code for specific amino acids, you know, when you're doing transfer RNA, like transfer RNA and so forth. But the genetic material and heredity is in the DNA, right? So deoxyribonucleic acid, it's ribose with no oxygen on it, with one oxygen molecule knocked off. Okay, and it's double-stranded. It's double-stranded, so you've got a strand of the nucleotides on one side, and then they're coiled. It's like a helix like that. Then you twist it like that, and then the bases will be touching like that. So it forms the genetic material of the cell. That's the DNA. It replicates itself, you know, when the cell divides, so that you have a set of the genetic material in one cell and a set of the genetic material in another cell. All right? So it ensures genetic continuity. It ensures that characteristic of life we talked about, which is, what is the characteristics of life that is associated with the function of DNA? What do you need DNA to do? What is it useful for? Reproduce. You can't reproduce if you don't have, you know, it's like the blueprint. See, like you can't build a house without the blueprint for the house, and you won't know what to put what. Same thing. So it really contributes to the characteristics of life in reproduction. All right? It also provides instructions for protein synthesis. When the cell, it instructs the cell to make a certain kind of protein, and then again, it affects the protein structure, which is going to affect the function. So each cell is specialized to make a certain kind of protein or a certain kind of enzyme, and then another cell is specialized to make, and all these cells make the tissues and the organs and so forth and so forth. And each of those systems is responsible for a specific function based on the kind of proteins that they make, the kind of enzymes that system makes. All right? So, contains the monosaccharide deoxyribose. All right? And so these are them: adenine, thymine, guanine, and cytosine. All right? So A and T will join together. Guanine and cytosine will be together. All right? Because one is one ring and the other one is two ring. So we go to ribonucleic acid. So note the differences on the RNA to the DNA, right? Because sometimes in the exam I ask you, what are the differences between RNA and DNA? Right? So the other one was double-stranded. This one is single-stranded. All right? Yeah, it's found in both the nucleus and the cytoplasm. Where do you find DNA? Only in the nucleus. You never find DNA in the cytoplasm. Why is that? Why is it contained in the nucleus? And the nucleus is a double envelope too. It's like double membranes on it. Why would you go to all those lengths? Yeah, to protect it, because I mean that's your blueprint right there. If it gets messed up, then you're going to produce the wrong proteins. You know, if there's a mutation in there, you're going to produce the wrong protein again. Okay? And then the protein is not going to be functional. You will lose functionality for that, whatever that protein does. Okay? The monosaccharide here is ribose. That's why it's called ribonucleic acid. The other one is deoxyribose, because you just knock off one oxygen on the ribose, all right, and then you form deoxyribose for DNA, and uses the organic acid uracil, all right, instead of the thymine. All right. All right, and messenger RNA, so it transcribes the message and takes the message out of the nucleus into the cytoplasm. And then transfer RNA is it's got 3 bases on it, on that transfer RNA, and 3 bases code for one amino acids like ACC or ATT or something like that. It will code for one of those 20 amino acids we talked about, so that it will affect the sequence in which the amino acids. So that's why the genetic material, if you just knock off one base, then everything is going to be off sequence. You see what I'm saying? And then all the proteins will be in the wrong place. I mean, all the amino acids, sorry, will be in the wrong place. And then because of the R groups are in the wrong place, it's not going to fold the way it's supposed to fold, and then the protein is not going to work. All right? And then ribosomal RNA is usually associated with ribosomes in the cytosol and makes proteins that the cell itself uses. The proteins that are made in the endoplasmic reticulum, when we start to talk of organelles, are usually going to be transported outside, because the endoplasmic reticulum is a membrane-bound organelle, right? All right. And this is the table with the functions, and you can go through this. All these functions, we went through them, like the structural, protein, protection for the lipids, and all those. We went through them, and this is just a good summary for all of them. So if you want to go through this, it will give you all the proteins right there. All right? And that's it. Thank you, guys. I appreciate it. We'll see you in a little bit.